Healing broken hearts. Scientists engineer muscle grafts to repair damaged hearts. Heart disease is a leading cause of death worldwide, and for many patients, the only hope is a heart transplant. But what if we could repair damaged hearts instead of replacing them? A groundbreaking study published in Nature on January 29, 2025, by Ahmad Fawad Gebran and a team of international researchers, brings us one step closer to that reality. Using engineered heart muscle, EHM, grafts made from stem cells, the team successfully repaired damaged hearts in primates, and even in a human patient. The study, led by researchers from the University Medical Center Göttingen and Stanford University, introduces a revolutionary approach to treating heart failure. By implanting lab-grown heart muscle patches onto damaged hearts, the team demonstrated that these grafts can improve heart function without causing dangerous side effects like arrhythmias or tumors. The researchers started with induced pluripotent stem cells, iPS cells, which can be reprogrammed into any cell type in the body. These cells were transformed into two key components: cardiomyocytes, heart muscle cells, and stromal cells, supportive tissue cells. These cells were then combined into a gel-like substance to create EHM patches. In a series of experiments, the team tested these patches in rhesus macaques, a species closely related to humans. Some monkeys had healthy hearts, while others had heart failure induced by a heart attack. The results were remarkable. The EHM grafts not only survived for up to 6 months, but also improved the heart's ability to pump blood. In monkeys with heart failure, the grafts enhanced the thickness and contractility of the damaged heart wall, leading to better overall heart function. The success in primates paved the way for human testing. In a first-of-its-kind clinical trial, a patient with advanced heart failure received an EHM graft. 3 months later, when the patient underwent a heart transplant, the researchers examined the original heart and found that the graft had successfully integrated into the tissue. The implanted heart cells were smaller and less mature than the patient's own cells, but they were functional and showed no signs of rejection. This breakthrough is a major step forward in regenerative medicine. Unlike traditional treatments that only manage symptoms, EHM grafts aim to repair the heart itself. The grafts are also safer than previous attempts at cell-based therapies, which often led to arrhythmias or tumor growth. While the results are promising, there are still hurdles to overcome. One challenge is ensuring long-term survival of the grafts. The study found that the implanted cells relied on anaerobic metabolism, a less efficient way of producing energy, until they developed a blood supply. Improving vascularization, the process of forming new blood vessels, will be key to making these grafts more effective. Another issue is the immune response. Even though the grafts were made from the patient's own cells, in the case of the human trial, the body still mounted a local immune reaction. Future research will focus on refining immunosuppressive treatments to prevent rejection while minimizing side effects. The implications of this research are enormous. For the millions of people living with heart failure, EHM grafts could offer a new lease on life. The technology could also reduce the need for heart transplants, which are limited by donor availability. Looking ahead, the team plans to expand their clinical trials and explore ways to optimize the grafts. Advances in 3D printing and tissue engineering could lead to even more sophisticated heart repair techniques. As Dr. Wolfram Hubertus Zimmermann, senior author of the study, put it, this is just the beginning. We're on the cusp of a new era in heart repair. So, while we're not quite at the point of growing entire hearts in a lab, this research shows that science is well on its way to mending broken hearts, one patch at a time.